Mushrooms in Mycoremediation with Willie Crosby. My name's Hannah. I am the Western Massachusetts Regenerative Food Systems Coordinator for NOFA Mass, and I will be hosting you today. I use she, her pronouns. I'll be going through all of this stuff here, kind of get the ins and outs, and then we'll jump into the content that Willie is going to provide us with today. So before we get started, I just want to take some time to acknowledge that we are presenting, attending, and hosting this workshop from land that was managed and inhabited before European colonization. Please take a moment. Christy will drop a link in the chat for you to find the land that you now occupy. It's a really cool map, actually. I highly recommend exploring it. And I join you here today from Puckamtuck land. A couple other ways, acknowledgement is one, and then also using our active selves in the racial equity work and process. A couple of ways we could do that together is by assisting BIPOC-led organizations such as the Urban Farming Institute, Gardening the Community down in Springfield, for example, African Alliance and Native American Rights Fund. By doing things like sharing resources, time, money, connections, kind of whatever we have to share with folks is super important as we move together forward in solidarity. It also includes supporting legislation that will begin the talk and advance the work towards reparations, as well as protecting the rights of food and farm workers who make a lot of this organic eating possible, right? And in the words of Chief Sequan Pikaji, also known as Chief George Spring Buffalo, he says, "We pay respect to the Massachusetts elders, past and present. We acknowledge the truth of violence perpetuated in the name of this country and make a commitment to uncovering that truth." So thank you today for taking that time to acknowledge the work and people and everything going on underneath the food system. Glad we can do it together. Just some session etiquette and info. Kind of the Zoom norms. We ask that everybody mute themselves. You can do that by pressing the microphone icon at the bottom of your screen. You can unmute yourself to ask a question, but we ask right when you do that to go ahead and remute just to avoid any background noise. You're welcome to use the chat feature and either both English or Spanish. We welcome questions. I will either field those to Willie throughout or at the end of the presentation. We have our translators here with us today. Thank you so much. So both our English and Spanish speakers have the option to ask questions. And please note that this session is being recorded. And of course, we have our sponsors. We could not do it without these sponsors here. Please take a minute to check out these people, these organizations, and make it all possible here today. Really couldn't do it without them. Feel free to support them, look for their products, acknowledge and thank them for their support. We also have our silver sponsors here. Again, lots of good folks who are helping to move this movement forward. And then we have our online auction. Christy will also drop that link in the chat for you. Again, lots of really great stuff like a 5-pound bag of coffee and some compost and all sorts of things. There's a microscope. So another really great way to support the presence of this auction or this conference here through auction, and lots of great things to look at, as well as our virtual vendors. So we have an online marketplace, if you will, for people to shop and kind of check out what our community has to offer. So please, feel free to check that out. And at this point, I will turn it over to Willie. Thanks so much for joining us today, Willie.
Yeah, it's my pleasure. And just curious, is everyone's audio good? If you just say yes or something in the chat, would appreciate that. Hannah's saying yes, so that's good.
You're good.
Yes.
Great. Yeah, I want to start us off with a breath coming together. If you feel comfortable, you can close your eyes and just feel your feet on the ground. And the beauty of this opportunity to be together the next hour, hour and a half or so, just this one time. So bringing your awareness to your breath, to the inhale and exhale through your nostrils. And just notice how your body feels. Notice if there's any movements or changes that you make when you start feeling your body, when you start listening to your body. And maybe bring into your mind something you're grateful for today. One thing, something that came out or you saw or it's just been on your mind. Yeah, what are you grateful for today? The one that comes up for me is crushes. So grateful for crushes. So welcome. Thanks for going into that. You can open your eyes and arrive back to this place. My name is Willie. I have been growing mushrooms for the last 7 years. I started a business called Fungi Ally back in 2014 in my basement, and started growing mushrooms. Got really inspired by Paul Stamets' book, Mycelium Running, and just dove in. So that made me excited to work with mushrooms, work with mycelium, and slowly over time it just grew and grew. We moved into a warehouse. New partners started to join the Mushroom Alliance, the Fungal Alliance, and we got up to the point in 2018 where we were growing about 500 pounds of mushrooms a week. And selling those to UMass and Smith College, kind of universities around here, and into Boston. So that was a really great experience diving into the commercial mushroom production realm. But like with a lot of commercial mushroom farming, it's very consuming. So I didn't have a lot of time for other aspects of like how mushrooms interested me, the medicinal, the nutritional, the mycoremediation, the education, these other aspects of mushrooms that continue to just expand and expand. So in 2019, I sold the fresh mushroom production part of my business, and have since really focused on research projects and education, and selling grow kits and spawn online to people that want to do different projects with mushrooms, whether that's growing them or doing mycoremediation or anything like that. So that's kind of been my winding journey to get here. And at this point, I'm just really excited about sharing my experience with mushrooms and my love of mycelium, and answering questions, and just going with people down the road of learning and exploring what fungi are and what they have to offer into our lives. And today I'm going to kind of focus on talking about mycoremediation and how mushrooms can plug into remediating earth, cleaning up pollutants, and making this a more desirable place to live in, since all these man-made catastrophes and pollutants. So I really believe the mushrooms and the mycelium are a really good ally to solve some of these problems that we have created. And I think it's important to note here that it's to solve for us. You know, it's not necessarily to like heal the planet or to save the earth or something. I think it's really for humans. If humans want to continue for many generations on this planet, we need to increase the tools and resources and allies that we have to keep it habitable. Yeah, habitable. So I think that's that's my outlook on it, and just wanted to share that. I'm gonna, Hannah, will you either make me a host or let me share a screen? And feel free at any point to ask questions. I have a chat box open so I can see questions and you can answer them as I'm going, and there will be little breaks as we go to ask questions. So always love going on tangents, whatever's on your mind. It doesn't have to be, you know, this or about mycoremediation or anything. Just stay involved, and that's fun. Okay. Yeah, I'm going to sprinkle in little facts too. Just sometimes nothing about mycoremediation, but number one fact is always store your mushrooms in paper bags. So they did a study in Poland, and what they found was that 80% of mushroom poisonings were actually from improper storage of mushrooms, not misidentification. So when you store mushrooms in a plastic bag, water builds up and they get moldy and bacteria grows on them, and then you get sick eating them. You store them in a paper bag, they just dry out over time. So store your mushrooms when you get them from a store or pick them in the wild in a paper bag in the fridge, and they'll keep for, you know, a week or two, and then they'll just be dried out and you can use them as dried mushrooms. So mycoremediation, when we break those two words up, we have myco meaning fungi and remediation mean like reversal of environmental damage. And mostly human caused. I'm sure there's places where it's not human caused, but most of the methodology of mycoremediation and focus has been human created pollutants. And I want to start with kind of this base layer of what are fungi and how do they grow, and just so we're all on this kind of baseline. And it does also kind of feed into how we approach mycoremediation and how we work with the mushrooms as we're trying to clean the area. So fungi are their own kingdom. They're completely separate than animals or plants. And there's three things that make a fungus a fungus. The first is that the cell walls are made out of chitin. And chitin is a really difficult molecule for us to digest. It's found in the cell walls of the mushrooms. And since we can't break it down, we need to break it down before we consume the mushrooms. So either by cooking or by hot water extraction or alcohol extraction, in some way you want to break down that chitin in order to access the medicinal or nutritional benefits of mushrooms. And this is just when you're consuming mushrooms. You know, this is like, we're talking about, instead of mycoremediation, like human, yeah, mycoremediation of the human body. So first thing makes a fungus, chitin. Second thing, their mode of digestion is absorption. So mycelium secretes enzymes outside of the body and breaks things down outside of the fungal body, and then sucks nutrients back in. So it's intended, instead of having this internal stomach like we have, fungi are secreting enzymes, digesting stuff, and then sucking nutrients in. And the third thing is they're heterotrophic, which means they rely on a food source here on Earth in order to continue to grow and exist and be. They uh, can't photosynthesize like plants do. So those are the three things that classify a fungus into that kingdom. And it's a really large kingdom, much bigger than plants or animals, anywhere from 1.5 to 8 million estimated species. So that's a huge, huge range. And we're still so new with what we know about fungi and how many they are and what they do. They're just amazing discoveries happening year after year. So it's a very rapidly evolving kingdom. And even now with DNA analysis, there's a lot of mushrooms that people thought were the same. Like European mycologists came to the US and were like, oh, it's the same mushroom. And we're finding out that it's actually different species. The European Ganoderma or reishi is different than the US reishi. So there's this big increase in species because of that. And of the estimated species, it's thought that about 140,000 actually produce mushrooms. And we know at this point, we know about 19 or 20,000 species of mushrooms. So we only know maybe 10 or 15% of the estimated mushroom producing fungi. Not all fungi produce mushrooms. So there's 13 phylums. As you go one step below kingdom is phylums. And there's 13 phylums, and only 2 of those phylums produce mushrooms. And a mushroom is a fruiting body that can be picked by hand and seen with the naked eye. So most fungal species don't have mushrooms. And that's okay when we're thinking about mycoremediation. And a lot of the other applications of fungi, the mushroom is really important. But with mycoremediation, the mushroom actually isn't an important part of the process. So we're not limited to working with just species that have mushrooms, but those are the species that we're most familiar with and that there's kind of the most amount of available mycelium to work with. And of the estimated 18,000 species, somewhere in the range of 1 to 10% are considered poisonous, and only 30 species are lethal, right? So that's less than the plant kingdom. So fungi get a bad rap for being dangerous, and people are scared to touch them or I don't know, get close to them in the woods. But they're totally safe to touch when you're in the woods. Any mushroom species you can touch, you can lick, you can even put in your mouth and just spit it out, and you won't get sick. So there's no need to be afraid of mushrooms or like keep 6 feet or anything from mushrooms. You can get up close, personal, smell them, taste them. That's encouraged from me. Just don't swallow. That's the biggest thing. And I want to go over the life cycle of one of those phylums, the Basidiomycota. And this is where most of the mushrooms that we consume come from. And they follow a pretty straightforward life cycle of you have your mushroom here, and it's producing spores. The spores go out and germinate, and they meet up, and they create mycelium. And the mycelium produces mushrooms, and it goes around and around and around like that. And it can vary time-wise. It can vary pretty largely. Like it can be a very fast growth cycle, maybe the course of a month, or it can be very slow over the course of years, for a mushroom to be ready to fruit again. So one of my teachers likes to call a mushroom a temple of copulation. It's where the sexual reproduction happens, and then genetically unique individuals, spores, leave the mushrooms, leave the gills. So those spores are produced on the gills here on the underside. This is a pink oyster mushroom. And the gills go out, or the spores go out, they leave the gills from rain or water or people or beetles, lots of different things. And they land on some sort of substrate and germinate. Here's what the spores look like. This was that oyster grow kit overnight. So there's a bunch of mushrooms all over the grow kit. This space here where it's not white is where the grow kit was sitting. And then all the white is the spores, right? So you can't see them as they're just coming off the mushroom. But if you leave it on a mushroom on a surface overnight, it creates what's called a spore print, which is all these genetically unique individuals that will go out and create mycelium and hopefully continue the life cycle. And they produce thousands and thousands and thousands of spores, right? So it's kind of like huge numbers game. Let's overwhelm the system with tons and tons of spores, and hopefully some will land somewhere they can grow and find a compatible mate. So at this point, they germinate, the spores germinate into mycelium, right? And they'll germinate whether they find a mate or not. Like if they find a suitable place to germinate, they'll grow out into mycelium, this kind of cottony white material. And they'll just start eating organic matter. So a lot of these saprophytes, which we'll talk about later, just consume dead organic matter. So like this leaf here. And mycelium only has one cell wall. It's very susceptible to drying out, or physically seems susceptible to invasion from other microorganisms. So you don't really just see it, as you walk around. You really have to dig into the ground or roll over a log or find the moisture. If you find moisture, you'll find mycelium. And when you see your bread go moldy, right, that's mycelium growing. That's just mycelium popping up on the bread and growing out. A very common contaminant in mushroom cultivation called trichoderma is typically what consumes bread as well. So from the mycelium, then we have the mushroom fruiting, right? And the mycelium is how the fungus spends most of its lifetime. So the mycelium is growing, expanding, finding new food sources, breaking matter down, excreting these enzymes. The enzymes are also like antibacterial, antiviral, even antifungal compounds to keep them safe, to keep other organisms from kind of coming into the food they're eating or coming in to attack them. So there's this huge array of chemicals and compounds, digestive enzymes that fungi and mycelium can produce to break things down. On the left here is a commonly used type of like medium for the mycelium to grow on for log cultivation. So it's called plug spawn, and you can just once that's myceliated, you can put that in the log, and it goes, it jumps from the plug onto the log. So that's our general life cycle, and I think the most important thing here is to know about mycelium, right? And just the what it can do, and that sometimes we're like, wow, especially right now, right? There's mushrooms everywhere. There's mushrooms popping up all over the place. And it's just because the environmental conditions are such that the mushrooms can fruit, but they're actually always there, right? They're always there in that mycelial stage, gathering food and kind of just like waiting until they have enough energy and there's the right parameters to fruit mushrooms. Because creating mushrooms is a very energy intensive thing for the mycelium. So it's not just like can do it all the time. Good. Okay, so any questions around life cycle and how mushrooms grow, anything in that realm? And feel free to type it in the chat box and I'll continue and I'll answer them if anyone puts something in.
I actually have a quick question. When you have the fruiting body of the mushroom, like the mushroom cap, let's say, how do you know when it's given its spore? I mean, I'm sure it's contingent upon the species, but like when it folds up, or like if you pick it before, has it already, like, is there a bad time to pick a mushroom? You know what I mean?
Yeah, so the mushroom, I don't know if you can see me, I think so, but like when it first fruits, this is the stem and this is the cap. When it first fruits, the cap is totally closed against the stem, and then it slowly opens up, and basically once you can see the gills, there's spores coming out, and then it flattens and then it flips up like that. So that's kind of the process of, and what it's doing is exposing the gills more and more. So as the gills are exposed more spores are leaving. But they're dropping spores at all times and more when they're flipped up like that. And for an edible purpose, you want to get them much earlier. You want to get them when they kind of have a curl on the margin typically. For cap and stem mushrooms, I like to say like a curl, especially shiitakes, a curl like your ear. The cap margin should have a curl like that. Like black trumpets, which are very prolific right now. It's pictured here in the center. Have a totally different life, like have a different form. So those ones, it's kind of like if they're dried out, they're no good, but if they're kind of decomposed. Yeah, and there's a lot of different like forms that mushrooms can take, ways that they disperse spores. So you have cap and stem, like gilled mushrooms. You have polypores, puffballs, which have the spores on the inside, boletes, which have tubes, clubs. So this is one of the kind of base identification features. Okay, what's the shape of it? How is it distributing its spores? And let's go from there. Okay. So looking at ecological roles, there's three kind of primary roles that fungi play in an ecosystem. We have up here in the top right, chaga, which is a parasite, black trumpets, which are mainly mycorrhizal. They might flip back and forth between saprophyte and mycorrhizal, and oysters, which are mainly saprophytic and can be a little bit parasitic as well. So parasites are an organism that attacks a living organism and depends on it in order to live. So chaga can't survive without the birch tree, right? And it's similar for like a true parasite, is they can't survive without their host. Now there's a lot of fungi that also become saprophytes, where they can eat a living host, and then when the host dies, they can continue to grow on that host. So like chicken of the woods is a really good example of that. And parasitic fungi are kind of where we run into a difficulty with fungi because of their ability to attack our food system. So, monocropping is very susceptible, especially when it's wet like this, to fungal disease. And that has really changed history over the years. Food impacts by fungi. I think I can share this video. Let me know if let's see if something's wrong. Oh, I think sound, share sound. Hopefully you can hear the sound on this as well. Where am I? Okay. Can you see this?
Yep.
Is there an audio?
No, no audio. Oh, wait, no. See, I'm not really sure how to make it work. It might be a setting, actually.
These bullet
Here you go.
ants have some worrying symptoms. Spores from a parasitic fungus called cordyceps have infiltrated their bodies and their minds. Its infected brain directs this ant upwards. Then, utterly disorientated, it grips a stem with its mandibles. Those afflicted that are discovered by the workers are quickly taken away and dumped far away from the colony. It seems extreme, but this is the reason why. Like something out of science fiction, the fruiting body of the cordyceps erupts from the ant's head. It can take 3 weeks to grow, and when finished, the deadly spores will burst from its tip. Then any ant in the vicinity will be in serious risk of death. The fungus is so virulent, it can wipe out whole colonies of ants. And it's not just ants that fall victim to this killer. There are literally thousands of different types of cordyceps fungi, and remarkably, each specializes on just one species.
Okay, I'm just gonna stop there. So that's a pretty wild mushroom, cordyceps. And one of the really interesting things about cordyceps is that there's an application. There's been several people who have been able to use that in order for pest control. So with both like carpenter ants and fire ants, there's kind of this biocontrol that cordyceps offer for colonies. So very interesting possibility there for pest control. Okay, so that's our parasites. Next up is mycorrhizal fungi. Mycorrhizal fungi, myco meaning mushroom, rhizal meaning root. So these are mushrooms that create a symbiotic relationship with plant roots. And basically the mycelium connects to the roots and expands out the area that the roots are covering. It also is much thinner. It's thinner than the roots, so it can get into little cracks and crevices and break down, rocks and get water that the roots otherwise couldn't access. And the plant is sending sugars down into the roots and trading that with the mycelium. So there's this exchange happening between the mycorrhizal fungus and the plant of mined minerals and water and sugars in form of photosynthetes. And the mycorrhizal fungi also connect individuals to create this one big web of mycelium that goes throughout the entire forest. So it's basically impossible now to really consider a forest to be filled with individual trees. It's very clear that there is just endless networks moving throughout a forest, and that the trees are all very well connected and aware in some senses. Like as they are dying, they are shifting resources to younger plants. As there is as there's like a new pest that comes in, there's alerts happening that these pests are arriving. Suzanne Simard is a really, really awesome, she gives a great TED talk, and I highly recommend. It might not be with 2 M's, I'm not sure. I highly recommend watching her TED talk and looking at the work that she has done on mycorrhizal fungi. Another cool stat for you is that every footstep we take, we step on 300 miles of mycelium. And most of this is mycorrhizal fungi. So if you take a footstep in the woods and you took that mycelium and laid it end to end, just in one straight line, it's like going from, I think, like Boston to Philadelphia or something. So it's huge, right? All that in one footstep. So the mycorrhizal fungi is very, very prevalent and extremely important to the health of the forest. Another cool book here by Michael Phillips, which is a little bit more about how to incorporate mycorrhizal fungi into the landscape and farming and having an orchard and that sort of thing. So another good resource in the mycorrhizal realm. And then our biggest allies, the saprophytes. These are the mushrooms that break down dead material. So they're the main reason that we're not just buried up above our heads in dead stuff, right? They're this link between death and new life. They take this dead organic matter and turn it into basic, simple building blocks for plants and other life to uptake. So they play a very, very crucial role in the recycling of nutrients and just the continuing of this whole planet Earth. And most of the common mushrooms that we grow and consume are saprophytes, primarily because it's much easier to use dead things, right? If you're trying to grow a mushroom, it's a lot easier to just say, okay, I need some organic matter, dead organic matter, than needing, with mycorrhizal fungi, you need to figure out a living host and the right soil biome and kind of all these right parameters. But with saprophytes, just great, get dead material and start the mushroom growing on that material. So these are the fungi that we grow, and they have evolved to break down lignin. So lignin is the second most common organic compound on the earth, right behind cellulose. And it's lignin and cellulose that comprise most plants, right? It's that combination. And what this is a complex hydrocarbon. Lignin's a complex hydrocarbon that these mushrooms, particularly white rot fungi, have evolved to break down. So it's these organisms that really are the best to work with when we're looking at cleaning up pollutants. So interestingly enough, oil and other petroleum products are also complex hydrocarbons, right? So anatomically or molecularly, I think molecularly is the correct word there. Molecularly, they are very, very similar, lignin to petroleum molecules. So we have these white rot fungi that have learned to digest lignin, that can be trained to also digest oil. And it's amazing in the lab. I was talking to Trad Cotter once, and he was saying how he was able to train the mycelium to eat 100% atrazine. So there's no other food source other than this really toxic pesticide, atrazine. And the way he was able to do that was starting with a normal Petri plate and adding in 5% atrazine. And slowly the mycelium, figured out how to break down atrazine. And then he increased it to 10% and took a culture, a little piece of mycelium from that 5% plate, and put it on the 10%. And then he did it again with 20%, then again with 50%, and again with 75%. And each time it would kind of stall, like the mycelium would be like, whoa, what is this? And then once it figured it out, it would just erupt and be like, oh, great, I can eat this. I'll keep going. And he was able to get to the point where he was growing 100 mycelium on basically just atrazine as a nutrient source, which is incredible, right? And the way that works is when I was talking about the mycelium's capacity to digest externally, they basically have this like keychain, like a janitor, a huge janitor keychain of keys. And that's the chemicals they can create in order to break down a compound. So when they interact or come across a new molecule or a new food source, it's kind of like this going through the keys and being like, how do I break this down? Is it this key? Is it this key? Is it this key? And once they find the way to break it down, there's kind of this like, hey, everybody, this is the key to use. And it's kind of off to the races. So there's this big catalog of compounds that mycelium can produce to help to break down these petroleum-based products. Some of the common commercial pollutants that there has been research on for breaking down is TNT, nerve gas, atrazine, other pesticides, oil. A fair amount of oil studies have been done, other petroleum products, and also bacteria. So filtering out and consuming bacteria, either in kind of like slow water or up and down water. I don't know. Not in submerged water situations, right? But like in flooding situations where there's an increase in water and then a decrease, possibility there. And curious here, some of the common at-home pollutants. So kind of thinking, oh, where would this be applicable just outside in your backyard? And curious if there's any others that I came up with that you are like, oh yeah, there's this thing. I guess light bulbs just came to my mind. I don't know. I don't think that's something that mushrooms can break down. But if there's any others, throw them out there. But these are all things that fungi can help to break down to very basic simple digestible things for other organisms. Yeah, paint or heavy metals. Fungi can't break down heavy metals. So this is the one where I think plants are actually much better because you can hyperaccumulate the heavy metals into plants and then remove them. With fungi, they do hyperaccumulate the heavy metals into the mushroom. So in the mushroom fruits, there's a ton of heavy metals in the mushroom, but it's not really practical because the mushroom comes and goes so quickly, right? You only have like 3 or 4 days to harvest the mushroom, whereas with the plant, it's there for months. So plants are a better approach for heavy metals. And it's interesting. The mushroom is also a kind of like a toilet in some way for the fungus, where when there's something that the mycelium doesn't want or is like, this isn't good for me, they'll actually put it into the mushroom. And so it's this way of getting it out of the mycelial network. So interesting way to look at a mushroom. And that's part of why it's like not recommended to eat mushrooms that are used in mycoremediation, right? I haven't seen testing of specific mushrooms that have been grown off of mycoremediated soil, but something I definitely wouldn't recommend doing. Yeah, bike grease, if you have a rag, car oil, being able to break that down. Food waste, water filtration, if there's any sort of gray water system or septic or composting system. Manure from animals and cardboard is a very, not really a pollutant, but easy thing to grow mycelium and build organic matter with mycelium. And that's one of the great things about growing mushrooms or doing mycoremediation is the end product is high quality organic matter. You're making good soil that worms can live in and thrive in and want to be in, and just building organic matter. So that's the best part of I think that's one of the great, eating mushrooms is great, but also that the end product is just increased organic matter is fantastic. Any questions up to this point around ecological roles or pollutants or anything like that? The next thing I'm going to jump into is 3 case studies looking at oil degradation. So we'll kind of see some of the mycelium at work in different trials. So the first one is probably the most commonly known trial that was done out in Washington, a partnership between Battelle Labs and Stamets, which they basically took there's 3 piles of motor oil or different oils in soil, and it was measured at 10,000 parts per million per gram, I believe. Usually it's measured in per gram, and they added tons and tons of spent oyster mushroom substrate. So that's just oyster mycelium on sawdust. And after 6 weeks, they covered it with a tarp. After 16 weeks, there was under 200 parts per million oil detected per gram, and that's a level that is possible to use. You can use in like a landscaping setting next to a highway or something. So this reduction rate is massive. Massive reduction, and a lot of that turned into mushroom material or organic matter. So here they have the this is, I think it's like 10 or 12 weeks after inoculation. And clearly the oysters are loving it, right? They are doing really well. They have huge fruiting bodies, and they just adapted into growing this oil substrate and went bonkers. And then this is at that 16-week period where you start to see plants coming in. And in the study, they had worms, and there was soil life, and even plants, and then birds coming in. And this is what remediation is, right? It's kind of going from this black pile of like hell, like nothing can grow, into slowly and steadily into plant life, soil that is supporting life. And the other two piles, they had a control pile and another sort of some aspect of remediation. Those two didn't do nearly as well. At this point, they didn't have any plants on them. There was no soil life. So the oyster mycelium was extremely important in the degradation of that oil. So that's first case study. This is second one, Fungi farm in California, looking at using oyster mycelium to clean up diesel. This, I think, is in the I'm spacing on the place up there. That's okay. So basically, there is behind a factory, there's a diesel and motor oil spills from generators and holding tanks, and there is really close to the river, maybe 40 or 50 feet from the river. So the company asked this company to come in and see if they could break down the diesel and motor oil to very low levels. They used oyster mushroom mycelium and did two applications, one in 2012 and one in 2013, and then tested in the following year in 2014. So this was a two-year growth period. And for both of these, the soil has been removed, right? So that's kind of one of the big pieces is one, removing the soil, and two, having enough organic matter to add in, right? So we're adding a lot of organic matter into these soils to bring them to life again. And the way of they found a really brilliant way of inoculating. Instead of, so the first time they used they tried to colonize straw, and the second time what they did is they just got huge rolls of burlap bags and pasteurized them, and then inoculated them with oyster spawn. So basically soaked them in water that was at 150, 170 degrees for 2 hours, and then sprinkled spawn onto them, put them into a plastic bag. You can see over here on the far right, and then the mycelium grew out. And then they can put, when they put those burlap bags out, they can put straw or wood chips or other organic material right onto that, and the mycelium jumps off and starts growing onto the new organic material. So a really ingenious way to inoculate and introduce mycelium into the remediation project. And I think really the best, like the most abundant source of mycelium that people usually have access to is local organic farms. So when mushroom farmers finish fruiting the mushrooms, they generally just throw the bags out, right? And it's on a big farm, literally tons of mushroom mycelium that can be used for these sorts of applications. So you don't always have to inoculate your own or grow your own mycelium. There is kind of waste streams of mycelium that I think can be tapped into in a lot of places, especially as mushroom farms have expanded in frequency and location. So this is a graph of motor oil concentrations, and the untreated here didn't really change much from the beginning, down to 250 milligrams per kilogram. The orange here is after the first year, so it's treated once after first year. And you can see it went down some on this one. On these two other samples, it went down significantly. And then once treated twice after 2 years, a very significant reduction in motor oil detection, right? So both of them are under that 50 threshold, 35 or 42 milligrams per kilogram. And that's a livable soil. You know, again, they found that a lot of worms were coming. There was plants starting to grow. It was kind of, this soil had come to life again. So another really good demonstration of oyster mycelium being able to break down motor oil. And here's a similar graph with diesel. So looking at the reduction of diesel in that same time period. Again, untreated, very high, 760 milligrams per kilogram. And then as we go down, year 1, much lower, these very low, and year 2, or 2 treatments, it's you know, more than tenfold less, huge decrease in detectable diesel in the soil. So that's to me, that's a really exciting application for mycelium. And you know, I'm sure around any car shop, mechanic place, even bike shop, you know, you have these very oil intensive either rags or spills or soil or anything that we can be breaking down and making into soil just by growing mycelium on them, you know, for a year or two. So very hopeful to me, you know. And who knows about applications on a large scale, right? There's I think that's some of the work and some of the questioning is, how does this scale up, and how are we capable of applying this to both in community, small scale, and up into larger scale spills and sites that are polluted with these sorts of things. How expensive is doing this type of remediation? That's like a big it depends question. If you're just buying mycelium, if you're buying like grow kits or something, it can get kind of expensive. But if you're getting mycelium just for free, or if you're expanding your own mycelium, it doesn't have to be very expensive at all, right? Instead of burlap, you could use cardboard. And cardboard's just basically free resource. So you could have cardboard, put spawn on that, mycelium grows out, and then add the cardboard into your contaminated soils, or vice versa, add the contaminated soils into your cardboard. So it doesn't have to be a super high-tech thing, right? This is very much doable backyard science. This third one was the same farm, that Fungi farm out in California. This was 2 years later. They kind of did a similar job at a different place. And what they did is they, again, the soil was contained in trash cans. It had been removed from the site, put into trash cans. And what they did was they layered the so they put plastic down, and then put down a layer of untreated straw, just straw. Put it down and wet it with a hose, and then put down a layer of mycelium, and then another layer of straw, and then the contaminated soil. Right, so they kind of did this lasagna method, layering straw, mycelium, straw, contaminated soil, and left that for 2 years. I think they reapplied again after the second year. So they did 2 treatments of this, and had again a massive, massive decrease in the amount of motor oil that was present in the soil, right? So going from 11,000 milligrams per kilogram all the way down to 230 milligrams per kilogram. So, and I'm guessing for another, if you did another, year, that would get down to close to that kind of 50-40 range, where it's fine to be used in like landscaping settings by a highway and somewhere where it's not necessarily for food. And you can also see that the diesel decreased significantly as well, going from 370 milligrams per kilogram down to 29, right? So so this mycelium is really doing a good job at breaking down material. All it needs is like another organic material to start growing out into. So either sawdust or wood chips. So we can start that growing process and then grow into the contaminated soils and break that material down. So 3 really good examples of, let's see if I can, oh, no. I can copy it from here. Cool. So this is the link to the last one that I was describing. It's just a 5-minute, really short video. I just put it in the chat box. Take a look at it. It's really great, and just kind of makes me be like, oh, this is straightforward to do. I don't quite understand why we don't do more of it. Just in our regular lives, like when we spill a little bit of oil or something, like we can just get a $20 bag of spawn and be making that into organic soil. That's good to support life. So easy solutions. And this is kind of like at-home version of that similar thing. This was a trial going on in a lab, so it was contaminated purposefully. But say you have a bike towel that you use a bunch that has grease on it, grease from the chain, or you change your own oil, and what do you do with it? Here you go. You can be growing out oyster mycelium, add it in to the straw that you are putting in the bucket, or just pour it on after the mycelium has grown out, and it'll just consume over the course of a year or 2 years, depending on how high the concentration is. It'll just consume that oil. So 5-gallon buckets are great to do this in, or those like 18-gallon totes, lots of different versions, anything that you can seal, right? So we want to be containing the pollutant and have a kind of moisture tight area for the mycelium to grow out and break material down. The top 2 mushrooms that are primarily used for this, I mean, the number one is oyster mushrooms, as we've seen all of these with petroleum products have been with oyster. And the second one is wine cap, and wine cap in particular for consuming bacteria. The mycelium is very ferocious in eating bacteria, and it's kind of social. It's like it likes to hang out with other organisms and see what they're doing and growing around them, and sometimes using them for food. So both these 2 are great mushroom producing fungi that you can buy mycelium for very easily, and can grow very easily, right? You can have a lot of mycelium on hand and use it for mycoremediation. And then, like I was saying at the beginning, there's many non-mushroom producing fungi that are very helpful in the mycoremediation world. They're just a little bit harder to study, right? Because we don't know, it's hard to know that they're there. And it's hard to start culturing them and getting them into a place where we can grow out and expand. So using these accessible mushrooms that people have been growing for a while and we know how to grow is a really helpful, is a really good starting point. So I want to talk about growing those two mushrooms, wine caps and oyster mushrooms, unless there's a question or a different direction that anyone wants to go at this point. So I will continue talking about growing wine caps. And wine caps, I think, is one of the best mushrooms to have just in your backyard, because once you have a patch established, you can just expand it and expand it and expand it. You know, you can expand from this wild mycelium, which is very unique to wine caps. So this is a good one to get established, and then you can, bulk up and expand and grow into all different places from where you have started it. The cap is this like wine burgundy red color. The bottom is gilled here, and it has purple to black spores. Typically at the bottom of the stem, it's holding on to material. So it's holding on to organic material and has these long threads of mycelium. Here you've got an egg to show size, how big these are. The partial veil here at the top, here, those are all, that's typically stays on the mushroom. So all key identification features. If you ever buy spawn from someone of this, then you can ask them and like double check, like, hey, is this wine cap? And send them the photo, and they'll be able to tell you. So for inoculating wine caps, lots and lots of different ways to do it. Here's just a very basic starting point. So you have a 4x4 area and get it down to bare ground. And this is with 1 5-pound bag of spawn. So, a 5-pound bag of spawn is like $30. And you'll be able to expand this out as much as you want to. And I would have about a half yard or a yard of wood chips to start out with. So you clear out this small 4x4 area. It can be, it's ideally in the shade. It can be in partial sun. It can be in full sun, but you just need to make the wood chips and sawdust much deeper. So best to do it in the shade. And then you put down a layer of sawdust or wood chips, about 1 inch or so. So you cover the ground. On top of that, you layer the spawn. So it's just like putting sprinkles on ice cream. You just break up the spawn into little pieces and spread it out. Mycelium has the incredible capacity to just regrow after it's been broken apart. So it's like if I could just cut my pinky off, go put it in a pile of wood chips, and then a month later, there'd be 10 of me, right? That's what mycelium can do, is just like you cut it into all these little tiny pieces and it just grows and reconnects and makes this one organism. So all this spawn is going to grow out and become one organism in this forest floor. So you lay out the spawn, and then on top of that, you put the wood chips. So these are hardwood, fresh wood chips. Has to be hardwood, maybe like at max 20% conifers, but mostly hardwood and under 1 month old. So when there's just wood chips hanging around, right, after 2 months or so, you have a bunch of mycelium in there. There's already other fungi that have started to grow. So with this, we want to kind of get ahead of those native fungi and get the wine cap established beforehand. So you put the wood chips on, and then they'll start fruiting about a year later, right? And after 6 months, that whole bed should be, let's see here. Yeah, that whole bed should be colonized. So if you dig down after 6 months, if you dig down into the wood chips, you'll see this thick, vibrant white mycelium. And instead of going back and buying spawn again, you can just take handfuls of those wood chips. I like to take like a 5-gallon bucket and then repeat the same thing, right? Just do a 10 by 10 area and expand it out. And take a 5-gallon bucket and add a fresh 5-gallon bucket back in. So it's nice to kind of have a patch where you're like, okay, this is my like mother patch. I'm just going to take and feed it mycelium, and I'm going to keep on inoculating all these new wood chip piles that I see around. And again, we're just building organic matter while producing fresh edible mushrooms. And these would be great for making bunker spawn, which is like if you fill burlap bags, right? If you fill burlap bags with the wood chips, the myceliated chips, or a 5-gallon bucket of myceliated chips and a 5-gallon bucket of unmyceliated chips, and put that into water runoff, right? So places, particularly where bacteria might be high, or places it's harder with it needing to be shaded, but where there's water running off of a parking lot. So like at UMass, for example, they've now over most of the parking lots, there's huge solar panels, right? So it's very shaded. So they could be where the water drains, they could have these filtration bunker spawn to filter the all the oil drips and all the stuff that's coming out of the cars that are parked in those parking lots, to filter through the mycelium before it goes into the sewer. And the mycelium would be catching some of those pollutants. So a great way to have mycelium and be ready to apply it as needed. This is another version. This is my mom that did a wine cap bed, and she kind of just sprinkled the spawn on top of beauty mulch type of stuff. And it was my Mother's Day present to her, and I told her, yeah, just have fresh wood chips, and she got beauty mulch, and I didn't have the heart to tell her it wouldn't work, so we just went ahead and did it. So put the spawn out, and then put an inch of wood chips or mulch on top of that, watered it in, and magically somehow there were mushrooms fruiting a year later. So I don't know how she did it, but they can sometimes do things that you don't expect them to do, and work in situations that you don't expect them to work in. Another setting of building a new garden with wine caps. So basically doing a sheet mulching where we have compost, newspaper, and cardboard, and then the spawn on top of the cardboard, and then sawdust and wood chips on top of that. You can plant directly into the compost right away. The plants grow up, create a shaded canopy for the mushrooms to fruit underneath. So, and those wood chips and sawdust break down in the course of a year to create really good, high quality organic material. Any questions around growing wine caps or their application into mycoremediation? Or in our last 10 minutes together, what I'm planning on talking about is growing oyster mushrooms.
Yeah, could you just repeat for me, Willie, clearing up, does the cap itself, I remember you saying that sometimes it holds the contaminants. Does it all do in the mycoremediation process? Does the cap or the fruiting body typically hold, or it kind of nulls it, eats it all up?
Yeah, it's so with the heavy metals, the heavy metals are hyperaccumulated into the
Into the fruit,
fruit
right?
into the mushroom. But with say motor oil or other petroleum products, it's just broken down. So it's broken down into basic carbon, oxygen molecules, and good to go.
Thanks.
But there's also like what else is in that? What else is in petroleum products? So it's better, definitely better not to eat those mushrooms. But it is broken down. It's not just like put into the mushrooms themselves. So some growing oyster mushrooms, an important part of all this is being able to grow the mushrooms if you are going to have the mycelium, unless you're lucky enough to get it all from a farm. The first step is substrate treatment with straw. So two common substrates. The first is wood chips. Wood chips you don't have to treat at all. Just like wine caps, just have them fresh and you'll be able to grow oyster mushrooms on them. The second common substrate is straw. And there's three primary different methods to prepare the straw for the mycelium to grow on. A lime treatment, which is soaking the straw in a lime water solution for 16 hours. Fermentation, which is soaking the straw just in water for 3 to 5 days. And basically it just gets really stinky. It's like when it's done, you kind of reach into the barrel and pull out a handful of straw and it's just like, whoo, this smells bad. That's when it's ready. And pasteurization, which is heating the straw, or this could be like in the case study that I showed, burlap sacks up to 150 degrees for about an hour and a half to 2 hours. So that's kind of our first step is treat the substrate, and then we'll inoculate. And inoculation doesn't have to be super clean or in a lab or anything. With these materials, you can just do it outside and not worry too much about contaminants. The big piece of inoculation is spawn rate. So how much mycelium are you adding into unmyceliated substrate? And one of the, typically in cultivation, we're working with somewhere between 5 to 20%. But if you do something like 50 or 60% myceliated material to 50 or 40% unmyceliated material, particularly with straw, you don't have to treat it at all, right? You could just kind of add that in, and there's enough mycelium that it'll take over that straw before other fungi come in. And also, when we're looking at mycoremediation, sometimes it's okay if other fungal species get in there. Eventually we want that to happen. We want more diversity in the soil, more organisms coming in and breaking down material. So it's okay if there is some contamination, but we do want a really healthy mycelial network to be growing into that contaminated soil to break down. One of the reasons oysters is used so commonly is it grows very quickly. So it's a really, really fast growing process. You can inoculate in 5-gallon buckets or plastic tubing or bins of any size, or just outside and use tarps. If you just have material and layer it in tarps and then just seal that up, that's a great way to inoculate as well. And layering unmyceliated material and myceliated materials the way to go. So that's one version of like inoculation table. Nothing fancy, right? Just outside with a bunch of grain jars and bags. And then the next step is incubation, letting the mycelium grow out. So this is typically takes a week to 2 weeks on small scale. On larger scales, it might take more like a month or so. and this is the process of the mycelium growing into that uncolonized material. And a important part in the mycoremediation world is where is this located, right? So if you have a big pile of material, you want to make sure that it's in the shade, right? And that it's in a cool space so it doesn't overheat. If it's, if it's getting blasted by the sun, even if it's wrapped in plastic, it's going to get really, really hot, especially if there's a lot of organic matter in the pile, it's going to get really, really hot. So you want to keep it in a cool place. Ideally, incubation does best in the 70s, right? So it's also, you can also think about what time of year are you inoculating. So probably for a large scale remediation project, inoculating in April would be the best. Like late March, April, maybe halfway into May, and then be like, okay, I'm not going to inoculate in the summer. I'll wait until September, October to inoculate. Right? If I was doing a big project that had to be outdoors and couldn't be kind of contained in a temperature setting. And definitely shade is very, very important, primarily so it doesn't dry out or overheat. And here's your example of what spent substrate from a farm would look like, right? So they're just throwing out either contaminated bags or bags that they've already fruited. So this is, if you go to a mushroom farm, you'll probably see a pile just like this. And all you need to do is remove those bags and you have that, a big brick of oyster spawn or mushroom spawn. And that can be broken up into little pieces and added into other organic material, either to remediate or to grow mushrooms themselves. So again, doing a 50-50 inoculation rate with these materials, the spent substrate, because it can be a little bit dirty, will really help for success. So if this is something you're interested in, I highly recommend contacting a local farm and just seeing, hey, is there a way to get a truckload of spent substrate? Sometimes they might ask you to pay $1 for a bag. You can also say, oh, just leave it. I'll just come, leave the bags on, and I'll take it and take care of it for you. Because this job isn't fun. Taking the bags off of the blocks and throwing them somewhere isn't really a good time. So contact your local mushroom farmer and see if they'll let you help them by taking their substrate, or if they see it that way. Thanks so much for coming tonight, talking mycoremediation. And last little tip is if you expose mushrooms when they're fresh or dried, gills up to the sun, especially this time of year, they convert ergosterol into vitamin D, and they store that vitamin D, right? So you can make these kind of vitamin D supercharged mushrooms by exposing them into the sun right now, and then just keeping them in a dark cupboard until winter. So thanks a lot. My name's Willie. My business is Fungi Ally. Feel free to reach out if you have any questions. I'll put my email in the chat box.
Great. Thanks so much, Willie. That's great.
Yeah, my pleasure, Hannah.
We have time for 1 or 2 questions. If anybody wants to unmute and ask. And then Willie has his, here we go. Yeah, has his email there, so you can reach out to him. All right, well, if there is no questions, I will wrap us up then. Yeah, that was so great. Thank you. And thanks to our interpreters for making this all possible, for all the folks who attended. And Christy has pasted us in the chat. Again, we have this most recent one she pasted is our survey. We would really appreciate if you fill out the survey. We're constantly trying to improve the way that we organize our workshops. So if you evaluate it, we can make this workshop better for you. Obviously, Willie did his basic talking NOFA mass, you know what I mean? And yeah, of course, check out our vendors again. Their support. We couldn't have done this without our vendors, and as well as the folks who supplied the auction. So feel free to click on those links. There's also a continuing ed piece. So if this is applicable to you, attendance to the NOFA Summer Conference can fulfill all 4 continuing education hours for AOLCPs. So if this applies to you, keep a record of your attendance, and in the next reaccreditation cycle, this can fulfill some of your credits. So remember to keep refreshing that program book link to make sure that your workshops are the most up to date. And we are looking forward to seeing you in the next couple of days.